So I want to go now to the next breakthrough or to the next excitement in brain research. And this is what I called, or what is called, the braibow technology. As you know, the brain, as you look at it, we call it the gray matter. It is gray matter. So when you look at it unstained, when you open the brain of any element, the tissue has the quality of gray or black or white. So you call it the white matter, the gray matter, but it's very, very difficult to see. So you can think about a jungle of trees, not stained as you saw before, and you see kind of a meshy thing, a brain, a gray matter. But now there is this new jump. A few years ago, a group from Harvard developed a molecular technique, a genetical technique, where they can really intervene with the genes, in this case of a mouse, particular genes, and they can locally insert pieces of DNA into the genome of this animal. There are many techniques to do it. This is the wizards, these are the miracles of molecular biology. They are able to really go into a genome and really manipulate it, engineer it, in order to add, in this case, a piece of a DNA. And when this piece of DNA is expressed during the expression of DNA, if the DNA is in a correct location, some cells, some cell types in the brain becomes colorful. So we can speak about the braibow, because suddenly the brain is glowing with colors, fluorescent colors. And so we see beautiful brains today, brain of mouse or animals, we can do it today. Of course, we don't do it to humans because you don't intervene with the embryo of humans, but you can do it to animals, and suddenly you get these fantastically beautiful braibow images, a colorful brain. So for example, this is a piece of a brain, not the cortex, but a piece that's called hippocampus. And suddenly you see the hippocampus, a particular region in the hippocampus, the CA1 region, glowing with colors. Each cell has, or different cells have different colors. Some are green, some are gray, purple, some are red, and some are yellow. And suddenly the brain is not anymore gray matter. It's a braibow brain. And this braibow brain enables you to do something fantastic. Because for the first time, at the level of light microscope, without cutting at the electron microscope, but at the light microscope level, you can just open the brain, look at it, and suddenly you see this, and if you zoom in more and more, you can start to see really the fibers, and you can see, at least at the first approximation, whether the green cell, so to speak, is touching the red, is going towards the red cell, so you get some kind of a gross anatomy. I will show you later that you cannot really say that there is a synapse, that there is a connection, yes or no, between this cell or another cell, because the light microscope resolution is not good enough to speak about synapses, which are smaller, too small to see in the light microscope. But still, you can get kind of a general first-order map, which is a very, 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 very important for modern anatomy. We are talking now about modern anatomy, unlike Ramon y Cajal and Golgi, which were the origin of anatomy. This is modern anatomy. It's a colorful anatomy at the light microscope level, and it's a connectomics anatomy at the EM level that I just showed you before. So this is a new technology, very useful for many, many purposes. We'll talk about what purposes are being used about that, but you can just stop for a second and enjoy the beauty of this technique. So this is part of the cortex. This was hippocampus. This is hippocampus, cortex, other regions in the brain, motor nerve ending that activate muscles. So you can see the beauty of it, and there are now a lot of exhibitions, art exhibitions, based on the braibow technology. So this is another contribution, not only to science, but also to the arts. So what can we do with the braibow that we could not do before? So here are some prospects or some examples for what you can do with this new technology. So for example, we are all interested for many, many years about the structural basis for learning. So now I'm talking to you, I hope you learn new things now. You didn't hear about the braibow before, maybe. Now you know something about the braibow. What happens in your brain when you learn? What is changing in your brain when you learn? So that means that you want to look at the brain in real time, online, when it is learning, now, when the mouse in the maze is finding his way from here to the cheese, and the second time he does it better, and the third time he does it better, what is changing in the brain when he learns? We can use this technology to look at the brain in real time, during learning, and suddenly you can see changes, anatomical changes related to learning. That's a breakthrough. In a black jungle or in a gray jungle, it would be very difficult. In a colorful jungle, it will be much easier to say that the green thing is moving or making contact or going there. So this is very useful as a technique to study online learning in the brain. Another use of this braibow technology is that you want to really start to tag, using these different dyes, different cell types. You want to ask a very basic question. How many cell types anatomically do I have in the brain? Is it 100? Is it 1,000? Is every cell different than another cell? How many types? So I can use these genetic tools to stain population of cells, different cell types, and say, okay, there are so many red cells, this one type, so many blue cells, another type. So I'm using this braibow tagging technology to characterize cell types in terms of anatomy. Very useful. So we know today much, much better how many cell types we have in the retina. It's not one cell type, but it's not million cell types. There are subtypes, subclasses. This is another use of the braibow. A third use of the braibow, and that's very important for us, neurobiologists, is to tag long-range connections. Because we know that some cells sense this long process, axons, sometimes from one region to another region in the brain. It's extremely difficult to detect this long path, very difficult, especially when it's a dark jungle. But when it's a red cell, you can really follow the red process, very long distances. And for the first time, we now know better where does this cell send its information, long range, not only locally. So this is also kind of a new advance due to a new technology. So you can see how technology enables us to ask old questions, but in a new way. So this was the second frontier. Let's go to the third frontier, brain-machine interface.